Okay, hi. I'm Jeff. This is joint work with John Osborne at UAB and other folks with UAB from here, but he's the official co-author. And what I wanted, this is, disclaimer, talk contains no medical advice. This is Don Mickey. He was one of the inspirations for when I was putting this stuff together. So he is colleague of Alan Turing at Bletchley Park during the war. I learned this very recently when I was actually at Bletchley Park, like a few weeks ago. Between 1955 and 1966, as a working geneticist, he has 13 papers in Nature. And in 1968, he creates Memoization, a concept that we still use today, in the journal Nature, no less, right? It's a very fascinating 4 pages. Some of it is, you can argue, not completely coherent, but I would argue that this still has very relevant bearing on what we do today. And his professional website, though he died in 2007, is still up, FYI, and there's some fascinating stuff on there. So I think, for example, this is the base class of LangChain. LangChain may eclipse PyTorch sometime soon in total number of users in the world based on downloads, and that's just kind of frightening. But what's in here is just large and surprising. And I would argue that if we read Mickey's paper correctly, maybe we wouldn't have done some of these things. So here's the outline of my talk. I want to talk about drug repurposing and what I'll call data repurposing to follow on that. I want to talk about the use case of Alzheimer's disease, drug repurposing, and then introduce the technique, put it all together, give a demo. All right, first thing, and these are all, apologies in advance, all of these, due to the interdisciplinary nature in here, everything is going to be oversimplified, and I'm so sorry, but this is just like my way of coping with the complexity here. So, very oversimplified FDA approval process. We go through a number of phases. As the number of individuals in the trial increases, we change our focus, initially from safety and eventually to efficacy for some specific disease. And success looks like putting a label on the drug that says it is specific, it has efficacy, it's been shown to have efficacy for some specific disease written on the label. So if you ever see use as directed, like what that actually means is a fascinating long story, but that I'm not going to get into. Here's a little primer if you want to understand what FDA sees as the top level summary, and then there's some fascinating other things that you can learn if you want to know more about that. Okay, so drug repurposing. Here's one definition, seems reasonable. Seeking new indications for an already approved drug rather than discovering a new de novo drug, because discovering new de novo drugs is ridiculously expensive. And if you can find a new application, sometimes that helps somebody who has no other way to be helped. And sometimes this involves additional wet lab study, and sometimes it can be done in a purely computational means. And here's a survey paper on different computational means of computational drug discovery, and tries to taxonomize things that people do. I'm just going to be able to pull one random story and use that as an example. Okay, so what am I going to call data repurposing? So data repurposing is sort of, I guess, maybe my personal philosophy on this. So I'm of the mind that having some data to inform a problem is better than having no data. And I don't want to say no to things because it's not structured enough or not trusted enough or not free enough. I feel like scientific advances typically happen when we can, sort of maximize along some of these dimensions jointly. And of course, all existing data is never really obtainable. There's only data that a team can find and use, which is a subset that we just is a limitation of being a human being. But typically, we're going to try as hard as we can to just push out to the edges adjacent to our problem. And maybe we do a small new thing. And that small thing, we could think of if we're including wet lab, it might have a time, we might be doing a functional study in an animal, but we might actually not need to do that. Maybe the small new data is something that we sit down and think about carefully and curate or call it something else. But that's sometimes where advances happen. So we might need to let go of some of this desiderata, even as much as we would want to have them, and just try to do our best to do science anyway. And so Here is a way that we can apply this to drug repurposing. For a given disease, we can find data related to that disease, like genes is a common characteristic of a disease, and I'll talk about an example of that soon. We can query gene-protein interaction data, and we can try to formulate an argument that an existing drug related to those two data sets might be beneficial for a given disease. Okay, example, Alzheimer's disease. Again, apologies, incredibly oversimplified. Here is a Alzheimer's disease sequencing project is a GWAS-based study where they compared people with Alzheimer's disease against a background of people who don't and found correlations. And they enhanced this with some of the standard things that you enhance these things with, linkage disequilibrium, EQTL, and some single expression, cell expression data. And they came up with a result list of 76 associated genes. Here's the paper if you want to know exactly what's in there. And what I'm going to say is that, this is, I'm picking this not because I want to be super deep dive on Alzheimer's disease. Is actually not what I study. Normally we do rare disease, and I work a lot with NLP, and that's not this, but this is a self-contained problem. And let me just comment on this little point set of evidence that we have on Alzheimer's disease is not really great compared to disease evidence that we have in other disease areas. So this is a disease, a genetic analysis in the area of obesity, which is has a stronger form of evidence. And so in this study, they were able to use a genome-wide association study to pinpoint a variant, single nucleotide, non-coding variant in this region over here, which actually regulates this thing way over here that's like 1.2 megabases away, and not this thing and not this other thing. And the way that they were able to do that is that they used a mouse model. And that is because a mouse model is really good for if you're trying to determine obesity, right? It's really easy phenotype to read out. Mouse models imitating human Alzheimer's disease, not as easy. And so it's just harder. So that's just kind of how it is right now with this disease area. It's kind of too bad, but that's just how it is. And so yeah, moving on. So here is something that I think is an improvement on the status quo. So I want to get work done, and I want to be efficient with my machine. And here's storage hierarchy of machine that I found on the internet, and it's got some main features on here, including some cost numbers, though I think they need to get updated a little bit, but relative-wise, I want to be really frugal on main memory and everything above, because I don't think the framework really should have opinions about what you're doing with your memory, because memory is scarce. Peter is building a 1,000,000-core machine. It's only going to have 2 terabytes of memory in it, right? Memory is not cheap. Let's not pretend that it is. So my library is going to use constant memory. It grows proportional to the stack size, very modestly. I want to modify the behavior of my system, I want to use the cheapest possible storage. So your disk drive, or if you have cloud storage and you want to use that, those things at the bottom. I want to relate those two things. And I'm also willing to gzip the data, because we usually have cores sitting around that make that easy. Okay, so here's my story. We're going to have everything so that we can be constant memory. We're going to have everything be a generator, a stream. And we're going to transform between that and a class called a block, an object. And there's going to be here I'm using Python notation. Again, broad interdisciplinary thing. I had multiple readers of the paper say, wow, everything great except for the Python part. And oh, I'm so glad this is grounded in the real world because we need to run Python. Can't please everybody all the time. Sorry, not sorry. So there's 2 implementations in the paper, one in Scheme and one in Python. I had to pick one notation for the slides in interest of time. Sorry, not sorry. So anyway, what can we do with this abstraction? We can transform in both directions, and then the down arrows are the action of apply. So we can take the class and apply it. It's actually sort of an object constructor, and then we can apply the generator, and the things on the left have metadata, the things on the right do not have metadata. And what this gives us is the ability to get 2 methods for one. The first method is the method that we would have just written if we were coding by our hands with streams. And the second method is a thing called signature. And the signature is going to basically consist of, you can think of it consisting of the method name, a version number, and all the arguments to the function, except not all the arguments necessarily, because that would be bad. So here is an expression in my sample program where I'm going to pull all the genes from the database that I'm using as a model, and I'm going to extract the NCB ID, the name and the ID from the file. And when I signature that, just the sub-expression genes is going to go look at all the things that I referred to and map those all in. And so some things we can see here is that it has some version numbers, some names of some arguments, but not all the arguments. For example, the data is not included anymore, right? I want to keep this thing to be a small thing, and that you can use for a couple different reasons. One is that it's going to make the computation much more efficient, and the other is it's actually useful human data to understand the provenance of the information you're looking at. So when you go back and you look at, like, why did this thing come out of my machine 2 years ago, you have as complete of a breadcrumb trail as you want to be able to go back and look at it. So let's see. Blanking on what I want to say about this. Right. So the API capability that you provide to the programmer through doing this is you have this discretionary ability to put .cached next to any sub-expression. And the behavior of .cached is to say, I want to use the resources in my machine differently. The default, if you don't say cached, is that everything is ephemeral, computed by the CPU every run, and it will just be thrown away at the end of the program. So you're not using memory, but you're using your CPU and your upstream data sources more. If you say .cached, everything will be automatically serialized to your disk. And if you run in that process or in a future process the same expression again, it will just automatically appear back without having been recomputed. And so here's an example of what this is not. So there are hundreds and hundreds and hundreds, an unimaginable number of build tools out there. And part of my curiosity of like writing this up was just like, can I explain why this happens so much? Like it's just a fascinating sociology. There's the common workflow language that was described earlier today, and they literally wrote themselves a web page with like more than 300 implementations, not even including anything even in the family that I'm describing. And then they wrote their own, like both a standard and multiple implementations. And it's like, wow, that's fascinating. What are people getting at here? So here is SnakeMake. I'm just picking it to pick on something like, whatever. There's a lot of things that I could choose, and I don't even know how to pick because there are so many. But SnakeMake is sort of elaboration on Unix make, as many of these things are. And this is how you concatenate two things together and stick it in a file. So I have a few objections to this. So like, what is that thing, the file? Like, how do you decide? Are you going to decide, how do you decide it's the same thing or different thing? Well, you're going to hash it. You're going to look at the timestamp. If it's a timestamp, which timestamp are you looking at? What does it mean? What if it's a remote artifact? If you write this file out, who's on the hook to parse it? Really, I don't want to think about any of these things. What I really want to think about is the stuff at the bottom that's like putting this thing together. So if you were in Python, the example thing, this can be any language, but this is what we have on the slides. You would write, take a list of generators, and for everything in the generator, we're going to take each one and yield it out, right? And then we can think about the thing that we want to think about, which is adding two things together. There's also a library function, so you don't have to write that. And maybe that block version number thing looks like a little bit of busy work, but that's actually a tool for the programmer to get to be able to decide how much of the system reruns. Which I'll get. My argument is the programmer can often decide better than the file system about which things need to be rerun. Okay, so this is a multilingual friendly protocol. In the paper, I go through two implementations, one in Python and one in Shea Scheme. And like every language has its own things, and I think that like that actually mimics sort of human behavior. Like we all have mental things. Some people have a larger capacity than others for the sort of like intrinsic complexity of C++. Like how would you ever love C++? I have a hard time understanding that, but like there's people out there that actually do, and they're just not like me. It's like one of these things of like, wow, that person is different than me, right? And I think that that's okay, and we can make less of a, we can have better collaboration if we can, do more idiomatic things in more different environments. So I'm not saying just rewrite it in language X. Okay, putting it all together. So I'm going to lock down here on this flexible term drug target that we have. And I'm going to say the one that I want to use is it's a specific protein believed to interact with a drug. Not something region in the protein and not a process, it's the protein. And so I'm going to formulate a metric for a drug, which is I'm going to use an NLP-derived gene-drug relation. I'm going to join to our ADSP gene list. And then for each drug, I'm going to compute this ratio of like how many times are these gene-drug interactions on target and how many times are they off target. Any questions about anything I've said so far before I keep going? Okay. So, demo. Okay, so here is the demo program, and this is everything in one file except for the library. And I'm going to start off here by changing how much of the data we're going to look at, which is like an idiomatic, part of this, writing this up is like understanding idiomatically what do you do to be productive. So right here is a number which I can change. If the number is 1, 1, that's the whole, use all the data. And what I'm going to say here is use 1/50th of the data. And I'm going to go over here and run, and I'm going to do one more thing here. Which is just stop. And then I'm going to run. And so this thing is going to just print me 3 examples of what's in my gene list. So we have PLCG2, TPCN1, and TSPAN14. And these are randomly selected things, of which I have asked 3. And the displayer that I wrote is actually part of the example code. It's not part of the framework. Uses reservoir sampling. And I'm going to keep moving down the list here. Notice that this is, of course, you'd expect something with, just a gene list to be fast. But like I'm going to move up here with the data quantity. And so here's a sample of 3 random nodes from the graph. And this is coming back in half a second because I asked it to only keep 2% of the data. And you would think that, so I can keep moving down. I make this argument in the paper, which confused some readers, that, this is sort of a partial substitute, economically speaking, with a REPL, right? Like the reason we use a REPL is because it's fast. Notice that I'm not using a REPL here, but it's still pretty darn fast. And when you're using a REPL, if you want this sort of like lots of dependencies between things, it's hard to get it to reevaluate just the stuff that you want. And so here's the sample of drugs as they appear in the database. And the database is RTXKG2, kindly provided by Steve and his team. Very useful resource. Again, this is a tool that's really not about knowledge graphs. Like I really want to just go forage data from anywhere. I'm using a KG because it's sort of like a really powerful shortcut to like, just show an example in the correct domain. And so here's, things that interact, ways that things interact molecularly. And these are, different kinds of relations that come out of the database, still, pretty fast. And now if I join them together, I can get the table that's in a table like what's in the paper. It's not going to have the same results in it as in the paper because we're only looking at 2% of the data. But qualitatively, you can see that this is kind of small on the screen. I'm sorry about that. Let me just pump that up real quick. Okay. So you can see by the fractions on the right, so it's printing out in the LaTeX that I just pasted into the paper, that it's doing the metric the way that I wanted it to do. And I can see that my program is working, and I only spent 4 seconds of computation time, 5 seconds. And then I'm going to pull, the last thing that I'm going to pull is a thing to drill into the top result to say, why are you telling me that? And so that's going to look a bit different. And every time I'm doing this, I'm actually computing all the results before again, right? But you know, it's still pretty fast. And so this one now comes back with like supporting text, like why are you telling me this? And you know, PMIDs and something that I can link all the way back to trying to make sense of like how does this information put together? At PMI, we take a great deal of care of like reading results and making mental sense of it, and before we communicate anything to anybody, and that's a really important part of the job. So finally, I'm going to go turn off the fractional sampling thing that I did above and just run the whole thing to get the result as I had in the paper with all the data. And that's going to run. It's going to do the first thing that makes the list is going to take about 45 seconds, and then the reasons is going to take about 30 seconds. Any questions about anything so far while that's running? Steve.
So this is leveraging the memoization framework as it.
That's right.
And that's what's enabling this to complete in 45 plus 30 seconds.
That's right.
Can you repeat the question?
Good, thank you. So Steve asks, this is leveraging memoization. Is this leveraging memoization? Is that why it's able to compute this so fast? The answer is yes and yes. If I were to drop my cache and just compute the whole thing from scratch, it runs in something like 12 minutes. Now, I could make it faster than that. What I have done is been super hard ass about like, it's literally the file that I pulled off of the translator server for Knowledge Graph Exchange, which has TGZ format, which means it's like not efficient, like, right? It's by design not efficient. It's to show that even if that's super not efficient to go pull your stream out, it's still like quite tolerable, right? So I'll show you what's in my file system actually here. Maybe right here. So so here is the RTX KG2C linked that doesn't have the file size, which is a little bit of a dissatisfying thing. So let me just do that too. So there we go, 4.5 gigabyte TGZ file, like clumsy, right? I mean, it seems like it should take work to do that, and somehow in 12 minutes I can get something useful out of it. On a laptop. And then the other interesting thing is if you look in the storage derived directory, you have the cached artifacts. They are pickled streams of pickled files, and the manifest files are human readable metadata for like where did this come from. So if you concatenate all of these manifest files together, you can just see each one says exactly what it is and what the signature was. So let's see if the other thing is finished by now. So here it is. I was doing some other things, so it took a few more seconds. Condoxatril and the reasons above. So like the reasons for why it appeared in the table. The table if you scroll up here before where it computed. So there's the table that appears in the paper. And then here is the reasons behind, and like it's not a bad. It's if you're gonna like drive some data, like this is a reasonable starting point, in my opinion. Like if you look at these PMIDs and what they're saying, it's like it's on the gene list, and like maybe you should think about that. And finally, scrolling in my history up to the. Where did I do? Come on. Okay, sorry, bash, So here's the scheme version, and that actually computed a result from a Python file. So it took the Git and SIG protocol, it sent it over Unix fork exec, and got the data, put it back into the runtime. So you don't have to have wars with your team about like which thing is in which runtime. You can just have everybody live in harmony and integrate and kumbaya. Questions? Oh wait, I had a couple things at the end that I just wanted to say. So this is a super tiny thing. It's 10 times smaller than the LNAI pipeline written by my friend Rondi Kinney was the system that made me aware of this specific form of memoization, and it's about 10 times larger. It has a little more code. It's written in Scala, and my implementations are 300 lines of Python, 233 lines of scheme. You can put it into your favorite environment with maybe in a day, so yeah, I'm not going to talk about that because of time. There's a few other things in the paper. And special thanks to Rodney, who made me aware of this specific memoization, Will, who encouraged me to write up the paper, and Steve, who provided the data source. Questions?
That was fantastic. One detail I couldn't quite remember from the paper, I apologize, is in the scheme implementation of the memoization and framework.
Yep.
What file format did you save as a S expression or something, or?
So you can do anything that your runtime knows how to round trip, right? And so in the scheme implementation, I used the read and write primitives. So in addition to those signatures being like human readable, the data is actually human readable as well. And you could also use, in Shea scheme, there's a thing called FASL, which is like fast serialization, binary serialization. The point is like each implementation gets to own its cached artifacts. And so like whatever makes that go, right? Like pickled streams make Python go, so there you have it. Yep.
Thanks.
Stupid question. What's a pickled stream?
It's a Python thing. Python comes. I don't know. Is it in the? Yeah, it's a built-in, right? There's a thing called a pickle. You can take any Python object and turn it into a stream of bytes, and vice versa. It's kind of janky. You don't want to save those on the internet and send them to your friends because it might actually propagate security hazards. But it's the Python thing to do. It's what all the cool Python kids do.
All right, thank you very much, Jeff. And we have, let's give him a hand.